If you talk about data description, so sources are same, and we have taken around 1200 samples where 12,000 samples where 6,000 were cancerous and 6,000 were healthy. And the motive of taking the balanced data set was actually because if we take a balanced data set, that will be very easy for a model to identify whether a particular sequencing is cancerous or not. And we are focusing on the stage 1 and stage 2 tumor. So we have taken the primary data set of that TGCA part. And technically we say that when we were training the model, we have taken 80% training part of this data, and remaining we were using for testing and even for the validation. Now this is some snapshot of the data set we have taken, and this is around 12,000 breast cancer DNA genomic sequence data we have taken, and all the genes ID has been mentioned over there. So ultimately there are having, I think, 12,000 rows and approximate 12,000 columns are there, and these all are the expression values we have taken. We have already the data we have taken, but the major task was to process the data. So this was a model architecture that we have taken. So the first task was that how to process the data, because the data was very imbalanced, and we have first of all balanced data. We have filled all the missing values by zeros and ones, and afterwards then we have proceeded with the sequence encoding. And in this sequence encoding, basically the DNA consists of ACTG. These are the variables which we use for the genomic series also. So we have taken all these protein, these are the, I think, nucleoids. All these protein sequences we have mathematically converted into an array consisting of that 100 sequence as we have taken, and then afterwards that particular data we have given to our CNN and BI LSTM model. Now there is a bifurcation of this model. CNN was basically working for the local part of, we can say, where there is a mutation in the data. If DNA is having some mutation in the local area only, then CNN is well capable of analyzing that part, that there is some mutation part going on, so that can be considered to be a tumor, or that can be considered to be something else, right? But if you talk about the BILST model, that deep learning model is basically learning the long sequence, means it is not analyzing actually a local, but it is analyzing a global sequence to analyze imbalanced pattern the genomic data is having. And then finally, our model, as we have taken a balanced data after the processing of one, our model is basically going to identify whether it is cancerous or not. And even then, we have taken some attention map technique that actually is available on deep learning, and even in Gen AI also we use that one. So that is basically used for the biomarker schemes, means we can then easily identify that if anyone is found to be cancerous, then what imbalance in the body is going on. So that actually is attention maps, which is working under the deep learning part. Now, talking about the training and optimization, so we have taken the ADAM. ADAM is basically an optimizer, which basically it is something like we revise. When we learn something, we revise. So that actually is the work of this ADAM tool, that library that we use. Learning rates were 0.0001, and we have trained our data in terms of 5 to 10 epochs. Now, talking about the matrices, so we have taken 5 of the parameters of deep learning. The first one was accuracy, precision, recall, F1 score, and TUC curve. So here we have the results and the comparison. So if you talk about, we have taken 3 of the model. The first one was, this is the table we have. The first one was, we have taken the machine learning unsupervised model, that is random forest, and then we have taken the CNN only from the deep learning, BILS came from the deep learning, and then what we have. So conclusion now. Conclusion says that as per my research and as per my implementation we have done, this model is working well with the approximate with the analysis with the prediction of 97%. But still there are some areas where we have to work on, and even our future work will be that even right now we have taken the genomic data of breast cancer, but there are some multiple sectors where we can work on. We will develop an integrated system which can even identify the other categories of the cancer using the genomic analysis. So this was my work. Thank you.